Hello everyone, welcome to today's session. My name is Dr. Caroline Morodi. Have you ever wondered what makes your blood red and keeps your body alive with oxygen? The answer is a tiny molecule called heme. And so in today's session, we'll take a journey through the heme proteins, looking at how the unique structures allow them to function, how the body makes them and breaks them down, and what happens when this delicate process is disrupted, leading to diseases. We will begin by examining the basic structure of heme, focusing on the periphery. Next, we look at the biosynthesis of heme, highlighting the key enzymes and regulation points. And then we move to heme degradation and bilirubin metabolism and associated clinical disorders. It's a naturally occurring derivative of porphyrin, and what makes it special is the ferrous ion sitting right at the center of the ring. Porphyrins are cyclic compounds. They are cyclic compounds. Think of them as a ring built from 4 little houses called pyrrole rings. And each pyrrole is numbered using Roman numerals, starting at the top, 1, 2, 3, and 4. These 4 pyrrole don't just sit, or rather don't just float around easily. They are linked together by methane bridges. And to make it even more systematic, each bridge has a Greek letter name. We have alpha, beta, gamma, and delta. Now, the porphyrin ring also carries side chains where different substitutes can be attached. These are numbered using Arabic numerals, and these are 1, 2, 3, 4, 5, 6, 7, and 8. In heme, we have 4 methyl side chains, 2 vinyl, and 2 propionate side chains, which are attached to the central tetrapyrrole. The ion is held at the center of the porphyrin ring, and this is done by nitrogen atoms from each pyrrole. And this ion is what gives heme its deep color and therefore its function. Where do we actually find heme in the body? Heme is not just sitting around on its own. It is always part of a protein, and depending on the protein, it takes on very different roles. In hemoglobin and myoglobin, heme is the oxygen carrier and storage unit. In cytochromes, that is P450, cytochrome B, C, and cytochrome oxidase, these heme proteins are involved in electron transport and energy production in the mitochondria. Cytochrome P450 also has a special role in drug metabolism and detoxification in the liver. In catalase and peroxidases, heme helps in breaking down hydrogen peroxide and therefore protecting the body against oxidative stress. So overall, heme is critical in respiration, detoxification, and also oxidative balance in our bodies. Now that we know what heme is and where it is found, let us look at how our body actually makes it. The main sites for heme synthesis are 1, the erythrocyte producing cell of the bone marrow, which produces about 80% of the total heme in the body. And the rate of synthesis in the erythrocyte depends on the rate of globin synthesis. We have the liver, which produces about 20%, and the rate of heme synthesis depends on the demand for heme proteins. Heme synthesis is not a single reaction, but a carefully ordered sequence of 8 enzymatic steps. The pathway takes place in 2 cellular components. Some reactions occur in the mitochondria and others in the cytosol. We begin inside the mitochondria where we have the rate limiting step. Then the pathway moves out to the cytosol where several intermediate modification occurs, and finally it returns to the mitochondria for the last transformation and crucial insertion of the ferrous ion. It is important to note that each step, it is catalyzed by specific enzyme, and any defect in one enzyme can block the pathway, leading to characteristic disorders called porphyrias, which we look at later on. Let's now walk through the pathway step by step from the very first condensation reaction all the way to the final formation of heme. In step number 1, we have formation of delta aminolevulonic acid. And this occurs in the mitochondria, and it involves a condensation reaction of glycine, a simple amino acid, and succinyl-CoA, a Krebs cycle intermediate, and the reaction is catalyzed by delta aminolevulonic acid synthase. This enzyme requires a cofactor, that is vitamin B6, and a deficiency in B6, or rather a deficiency in vitamin B6, will result to what we call sideroblastic anemia. The delta aminolevulonic acid formed in the mitochondria then moves out into the cytosol for the second steps, where 2 molecules of delta aminolevulonic acid condense to form porphobilinogen. And this reaction is catalyzed by a zinc-containing enzyme known as ALA dehydratase, also known as porphobilinogen synthase. This enzyme is extremely sensitive to inhibition by heavy metals, including lead, which tend to replace the zinc. This inhibition is in part responsible for the elevation in delta aminolevulonic acid and anemia seen in lead poisoning. In the third step, we have formation of hydroxymidylbilane, which occurs in the cytosol and involves condensation of 4-porphobilinogen, producing a linear intermediate tetrapyrrole known as hydroxymidylbilane. And this reaction is catalyzed by porphobilinogen deaminase, also known as hydroxymidylbilane synthase, or uroporphyrogen 1 synthase. Step number 4, we see the linear chain, that is the hydroxymidylbilane folds and cyclizes to form a ring known as uroporphyrogen 3. Structurally, this is a first closed tetrapyrrole ring, and at this point, the basic porphyrin skeleton is established, although it carries a lot of acetate and propionate side chains. This reaction is characterized by uroporphyrogen 3 synthase. In the fifth reaction, we have formation of coproporphyrogen 3. Here we see that the uroporphyrogen 3 undergoes decarboxylation, that is the removal of 4 carbon dioxide molecules due to the decarboxylation of the acetate group, generating what is known as coproporphyrogen 3. Structurally, this increases conjugation. More double bonds are formed, moving the compound closer to the fully conjugated porphyrin system. The enzyme that catalyzes this reaction is known as uroporphyrogen decarboxylase. The sixth step is the formation of protoporphyrin. Coproporphyrogen 3 enters the mitochondrion, and 2 propionate side chains are decarboxylated. The 2 propionate side chains are decarboxylated to form protoporphyrogen 9. And the enzyme that catalyzes this reaction is known as coproporphyrogen 3 oxidase. Protoporphyrogen 9 undergoes further oxidation to form protoporphyrin 9, and this reaction is characterized by protoporphyrogen oxidase. In the final step, we have formation of heme, and here we have a spontaneous introduction of the ferrous ion to protoporphyrin 9. And this rate of reaction is enhanced by ferrochelatase, an enzyme which is inhibited by lead, as we shall see. So, this is a critical step that transforms propofirine into heme, the biologically prosthetic group. Let's now connect the heme synthesis pathway to the clinical relevance. This is where it gets interesting. When there is a deficiency of iron, it will result to anemia. Therefore, there will be no complete heme synthesis. Lead poisoning also inhibits some enzyme in the pathway, including the delta aminolevulonic acid. This is a first step, which catalyzes the first step of the pathway. It also inhibits ALA dehydratase and ferrochelatase, which is the enzyme catalyzing the final reaction of the pathway. Lead poisoning, therefore, will cause a reduction of heme synthesis, resulting to microcytic anemia. When one of the enzymes in the pathway does not work properly, we'll get a family of disorders known as porphyrias. And this results to accumulation of intermediates, as we shall see in the later slides. So let us look at porphyrias in detail. And porphyrias are rare, inherited, and acquired defects in heme synthesis, which result in accumulation and excretion of porphyrins in urine and feces. Porphyrias refer to red-blue color caused by pigments like porphyrins in the urine of some patients who have defects in heme synthesis. When we classify porphyrins, we often do so based on the primary site of expression of the enzyme defect. Broadly, they fall into two categories. We have the erythropoietic porphyrias, where the defect is in the bone marrow, and we have the hepatic porphyrias, where we have enzyme deficiency occurring in the liver. Let's start with the erythropoietic porphyrias. One of the most important porphyria under this are the congenital or hereditary erythropoietic porphyria. And this is due to the deficiency of uroporphyrinogen 3 synthase. And this disorder is a rare autosomal recessive disorder. It results to accumulation and excretion of large amounts of uroporphyrinogen 1 and coproporphyrinogen 1 in urine. It's also important to note that high levels uroporphyrin 1 in erythrocyte will result to premature destruction of the RBCs. Clinical symptoms associated with these disorders include photosensitivity, hemolytic anemia, and also the bones and teeth may gain a pink color. The erythropoietic protoporphyria is as a result of partial activity of ferrochelatase, and this results to accumulation and excretion of protoporphyrin 9 in feces. Clinical symptoms include photosensitivity, which is non-blistering. Under hepatic porphyrias, we have acute intermittent porphyria, and this is caused by partial deficiency of uroporphyrinogen 1 synthase. It will result to accumulation of PPG and delta aminolevulonic acid in liver, plasma, and it will be excreted in urine. Clinical symptoms associated with acute intermittent porphyria includes abdominal pain, smooth muscle spasm, and constipation and hypertension. It's also important to know that disease occurs after puberty, and barbiturates, steroids, and alcohol can worsen this condition. The other form of hepatic porphyria is a variegated porphyria, and this is due to the deficiency of protoporphyrinogen oxidase. Patients with this disorder will excrete large quantities of PPG, ALA, and uro and coproporphyrins in urine and in feces. Clinical symptoms include photosensitivity and other symptoms may vary, and hence the name variegator. It's also important to note that alcohol and other drugs can aggravate or worsen this condition. We also have porphyria cutanea tarda, which is the most common porphyria, and it's caused by partial deficiency of uroporphyrinogen decarboxylase. And this will lead to excretion of large quantities of uroporphyrin 1 and 3 in urine. And the most common clinical symptom of this disease is photosensitivity. We have what we call acquired or toxic porphyria, and this is due to certain poisons or toxins and drugs, including heavy metal and alcohol. We also have drugs such as steroids, oral contraceptives, and pesticide, which result to acquired porphyria. Finally, we have the hereditary coproporphyria, and this is due to the partial deficiency of coproporphyrinogen 3 oxidase. And this results to large excretion amount of coproporphyrinogen 3 in urine and feces. We'll also see that PPG excretion in urine will increase in this condition. And the most common clinical symptom is photosensitivity. Here is a summary of the heme synthesis pathway together with various porphyria associated with this pathway. So as we said earlier, some reaction or some steps will occur in the mitochondria and also the cytosol. Now, it is important to note that if the defects are found in the liver, the patient will tend to have abdominal pain and neurological symptoms. On the other hand, if the defect is found in the bone marrow, patient will present with symptoms of sun sensitivity, skin blistering, and the red urine, but they will not have neurological signs. So having known about porphyria, it's important to note that they may seem rare, but they are a perfect reminder that when one enzyme slips in a long biochemical pathway, the consequences can ripple through the whole body, from the brain to the skin and the liver. And so as a medic or a biochemist, your job is to follow these clues, trace the pathway, and know where the traffic jam happened. All right, now that you have explored heme synthesis and the porphyrias, the next step is to ask, what happens to heme once it has served its purpose? We do not keep heme around forever, or rather the body does not keep the heme around. The heme released from the heme protein cannot just be left free in circulation because free heme is toxic. It generates free radical and damaged tissue. So the body has a well-organized pathway from heme degradation to safe disposal. So heme degradation occurs in the liver, and heme is degraded to bilirubin by the reticuloendothelial cells. In the first steps, it's a conversion of heme to biliverdin, but it occurs through a series of 2 steps. And these 2 steps, the enzyme that catalyzes this reaction is known as the macrosomal heme oxygenase system. So the enzyme binds heme in the presence of NADPH and an oxygen molecule. Here it specifically hydroxylates the alpha-methane bridges, and this will produce hydroxyheme. The hydroxylated bridge, that is the alpha, undergoes further oxidation, forming vandohemine. And this involves insertion of an oxygen molecule into the bridge, releasing 1 molecule of carbon monoxide. Vandohemine is further oxidized. Here it completely loses its ferrous ion and it opens up to form a linear tetrapyrrole known as biliverdin. Next, biliverdin reductase reduces biliverdin to bilirubin in presence of NADPH. And so the bilirubin formed is water insoluble, and therefore, for it to be transported or to travel through the bloodstream, it is tightly bound to albumin. So here we have an example of one of the heme protein, that is hemoglobin, undergoing degradation in the spleen. And so we have formation of bilirubin, which is a water-insoluble molecule. And so this bilirubin will be bound to albumin, and here it is transported via blood to the liver. In the liver, bilirubin is taken up by hepatocyte and conjugated with 2 molecules of glucuronic acid. And this reaction is characterized by UDP glucuronyl transferase, forming bilirubin diglucuronide, which is a soluble molecule. This is also known as direct bilirubin or conjugated bilirubin. So the conjugated bilirubin is secreted into the bile, stored in the gallbladder, and released into the intestine. In the intestine, gut bacteria act on it, converting it to urobilinogen. And majority of the urobilinogen is converted to stacobilin. It is converted to stacobilin, which is excreted in the feces and gives stool its characteristic brown color. Some urobilinogen will be reabsorbed into the blood and taken to the kidney as urobilin. So they'll be excreted in the urine as urobilin, and this is urobilin, and this is what gives urine its yellow color. So we have types of bilirubin. We have the direct bilirubin. This is a conjugated one, which is water soluble, and its reference range is between 0.0 to 0.2 milligram per deciliter. We also have the indirect bilirubin, which is unconjugated, water insoluble, and its main reference range is between 0.2 to 0.8 milligrams per deciliter. So both direct and indirect bilirubin will give us what is called the total bilirubin, and its reference range is between 0.2 to 1 milligram per deciliter. Clinically, defects or obstruction in heme degradation will lead to jaundice, which is a major clinical significance of heme degradation. It is important to note that the normal blood plasma bilirubin is about 1 milligram per deciliter. And in case of jaundice, the plasma bilirubin tends to be high. And this will diffuse into tissues, including the skin and the scale of the eye, and turns them yellow, a condition known as hyperbilirubinemia. We have various forms of jaundice, including prehepatic, hepatic, post-hepatic, and also neonatal jaundice. Now we look at each of these forms of jaundice. For prehepatic, also known as hemolytic jaundice, and this is as a result of excessive breakdown of erythrocyte. And this leads to increased production of bilirubin, and the hepatocytes will not be able to conjugate all the bilirubin formed. And therefore, it will result to elevation of unconjugated bilirubin in the plasma. When you talk about the hepatic or hepatocellular jaundice, and this is as a result of damaged hepatocytes. And so the site of defect is within the liver. So we'll find that the hepatocytes are either damaged or their enzymes are defect, or the enzyme is defective. That is, UDP glucuronidase transferase is defective. And we find that poisons, including chloroform, antibiotics, and hepatitis virus, can cause damage to the liver, and also cirrhosis will also cause damage to the liver. And so therefore, the hepatocyte will not be able to conjugate or even secrete bilirubin. And this will result to an elevated levels of unconjugated bilirubin in plasma. Post-hepatic or obstructive jaundice, and this happens after the liver, and it's due to the obstruction of the bile duct, and this could be as a result of gallstones and cancer of pancreas. And therefore, due to the blockage of the bile duct, conjugated bilirubin secreted by the liver returns to blood, thereby elevating levels of conjugated bilirubin in the plasma. Finally, we have the jaundice in newborns, that is neonatal jaundice. And most newborns show a rise in unconjugated bilirubin in the first postnatal week. And this is as a result of low activity of the hepatic bilirubin UDP glucuronidase transferase at birth. And therefore, it will result to elevated levels of unconjugated bilirubin, which will diffuse into the basal ganglia, causing toxicity and pathological jaundice. Treatment for this kind of jaundice is through phototherapy, which converts unconjugated bilirubin into water-soluble isomers that can be excreted. With this, we've come to the end of this session. We've looked at heme proteins, their synthesis, degradation, and associated disorders. In the next session, we're going to look at examples of heme proteins, include hemoglobin and myoglobin. Thank you.